Okay, we'll make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be finishing off cell membranes. We're not going to be looking at exam questions in today's lesson. We're going to do a full exam question lesson in our next week's lesson, where we combine everything we've looked at in our last lesson and today's lesson and put into sort of application question style exam questions. Today, what we're going to do is we're going to finish off the final part of the spec. So last lesson, we looked at the structure of cell membranes, what cell membranes have within them, and why they have those specific functions. We also looked at diffusion, facilitated diffusion, and osmosis. Today, we're going to look at active transport and co-transport, and that will finish off everything you need to know about cell membranes. Okay, we may finish a bit early today, but it's only a bit of content that we need to get through before we finish off this topic. So if we make a start again, here's the specification. In terms of what we did last lesson, we did everything in terms of um, we did all of this over here. This was your content that you needed to know. And we looked at simple diffusion, we did facilitated diffusion, and we did osmosis. Today we're going to do the active transport and co-transport, and we will also look at this at the end. I don't have a specific slide, but this is just content specific that you'll be required to know, which we will look at. Okay, and then this is basically your application exam questions that we will look at in our next lesson. So if we make a start on today's lesson, we're going to start off by looking at active transport. Okay, and at any point, if you have any questions, you can let me know. um that's completely fine. So there's a process to active transport, and I'm going to go through the process first, and then I'm going to come back to this image that I have over here to explain it in a bit more detail. So active transport, now if you think back to GCSE, active transport was the movement of sugars from an area of low concentration to an area of high concentration, um, from against the concentration gradient, and it requires energy in order for active transport to occur. Now, that still stands. We still use active transport for that purpose at A level. Now, in terms of active transport, however, we specifically focus on the energy aspect of it. So we know that active transport is against the concentration gradient, and we know that it requires energy. Now, if we think back to biological molecules, we learned about ATP, and ATP was your energy source, and we know that ATP is hydrolyzed into your ADP and your inorganic phosphate group, and that's what releases the energy for biological processes to occur. So that's basically, we need to combine that knowledge into how the ATP provides the energy needed for active transport. So the first step, we've looked at the cell membrane, and we know that there are many sort of different sort of methods that things can move across a cell membrane, and one of those methods are through carrier proteins, such as channel proteins. So if we have a look at this image over here, you can see that we have our phospholipid bilayer. This is a part of the membrane, and we have a phospholipid bilayer. This is your basically, these are your phospholipids. They are lining up, so your hydrophobic tails point inwards and your hydrophilic heads are on the outside. Okay, so you have your um, plasma membrane there, and you have your phospholipid bilayer. And in the middle of your phospholipid bilayers, you have these things over here, and these are called these things over here, and these are called channel proteins. And these channel proteins are basically involved in active transport. And the reason why they are involved in active transport is the following. So we know that active transport can occur through carrier proteins or channel proteins. Channel proteins are the things which span the whole length of the membrane. So what happens? The molecule which needs to move across the membrane will bind to a receptor complementary in shape on the protein. So if we think back to this diagram over here, we have a channel protein, and that channel protein will have a receptor. Okay, so we have, let's say on this side over here, what's going to happen is that the molecule is going to bind to the receptor. So we've got the extracellular fluid, which is on the outside, and then we've got the cytoplasm, which is on the inside. So this is the inside of the cell. The bottom half is the inside of the cell. So how do things come into the cell and how do things move out of the cell? Well, in order to move things into the cell, the molecule binds to a complementary receptor on the cell membrane. Okay, so for example, if this molecule needs to move across, it will bind to a receptor on the surface of that cell membrane first. Once it's bound to that receptor on the surface of the cell membrane, it needs energy to move across. Because if we think about it, there are more particles, more of these red circles inside the cell compared to outside the cell. So it needs energy for it to move across. So how does it do this? Well, the molecule first of all binds to the receptor so it can move into the cell. So it binds onto the receptor on the outside of the membrane, the complementary receptor. So for example, we could have sodium ions. Sodium ions might want to move into the cell. Okay, because sodium ions might want to move into the cell, they will bind to a receptor on the outside of the cell. Okay, now because there's a higher concentration of sodium ions inside the cell, we can't move by facilitated diffusion. We can't move by osmosis. We can't move by diffusion, simple diffusion. We're going to have to move by active transport because there's a lower concentration on the outside and a higher concentration on the inside. So we need energy. And at A level, that energy is known as ATP. So whenever you're mentioning active transport, always mention ATP as well. So ATP is going to bind to the carrier protein inside the cell and is hydrolyzed into ADP and the phosphate. The same thing that we looked at in biological molecules. So the ATP is going to be hydrolyzed into ADP, adenosine diphosphate, and an inorganic phosphate. That's what's going to happen. And when this happens, this releases a lot of energy. So releases a lot of energy. Okay, so what's happened so far? A molecule has come to a cell membrane and it needs to enter the cell. The molecule binds to the receptor on the outside of the cell membrane, and then it will need to move into the cell membrane. So what it does, because it has a lower concentration on the outside compared to the inside, it's going to get an ATP molecule. And that ATP molecule is also going to bind to the inside of the cell. So the molecule has bound to the outside of the plasma membrane. An ATP molecule has also bound to the inside of the cell. And that ATP molecule is hydrolyzed, okay, which means that it releases energy. It's hydrolyzed into ADP and a phosphate. So it's released a lot of energy. And what happens is because that energy has been released, the protein changes shape, and then the molecule which was bound to the receptor is absorbed into the cell and it moves inside um, into the cell. Okay, so that hydrolysis of ATP into ADP has released the energy for the protein, the carrier protein, to change shape and for that molecule to move into the cell. Because of the change shape, the molecule is able, a small gap is created, and that's what has allowed the molecule to move into the cell. Okay, now that phosphate ion is released. Okay, so remember, we're forming that phosphate ion. Okay, so when that ATP is converted into ADP and an inorganic phosphate, when that phosphate is bound to the membrane, that's what releases the energy. This phosphate is the energy releasing thing. Okay, when ATP is hydrolyzed and the phosphate is released, that phosphate binds onto the membrane and that causes the change in the structure of the membrane, causing the molecule to be released on the other side. Okay, and when that phosphate has done its job and released the energy, the phosphate is then released, and because the phosphate detaches from the membrane returns back to its original structure. The protein returns to its original shape, so nothing else can move into the membrane. So it's all because of this energy that active transport is able to happen through. So going over it once again, if we go back over here onto this image and we focus on this molecule over here, what's going to happen is that this molecule binds to a receptor. Once it's bound to a receptor, it's then going to tell an ATP molecule to also bind to the inside of this protein. Okay, so it's going to call an ATP molecule to bind to the inside of the membrane. Now, what the ATP is going to do, this ATP is going to be hydrolyzed into ADP and PI. So ATP converts into ADP and PI. And when it converts into your ADP and PI, this phosphate group is going to attach onto the membrane. And because that phosphate group is very powerful, okay, it will cause the membrane to change shape and open up. Okay, so the phosphate binds to protein, and that's the thing which releases the energy. Okay, so it releases the energy, causing the membrane to open up, the protein to open up, causing a change in shape. And that change in shape forces this molecule to move across into the cell. Now, once it's moved across and the phosphate has used up all its energy, the phosphate over here will detach from the protein, the membrane protein that we have, and it will go and combine with your ADP again to reform your ATP. And because the phosphate has left the protein, the carrier channel protein, the protein can then return back to its original shape. That's what happens in active transport. Okay? And that's how things are absorbed through active transport. The principles that you learned at GCSE are still the same. It's still moving from an area of low concentration to an area of high concentration against the concentration gradient, and it requires energy. All of that is still the same. However, we get a bit more technical in where that energy is coming from, and we focus on ATP. Molecule binds to a receptor. ATP binds onto the inside of the protein, the carrier protein or the channel protein. ATP is hydrolyzed into ADP and phosphate. Phosphate attaches to protein, causing a change in shape, releasing the energy. That release of energy will cause a change in the shape of the carrier protein, causing the molecule which is attached to be released on the inside of the cell. The phosphate then detaches from the carrier protein and the protein returns back to its original shape. That's your active transport. Okay? If you have any questions on active transport, please let me know what we're going to do now is we're going to move on to co-transport. And co-transport is basically a type of active transport which you explicitly need to know about. Okay, and this is your co-transport. Okay, and again, what I'm going to do is I'm going to go through the steps and I'm going to link to this diagram every now and then. So, co-transport, again, it's a type of active transport. Okay, we're going to go through it because this is an aspect that people find a bit confusing, because there are a lot of steps and there's model exam answer that you can be asked for co-transport, which we'll look at in our next lesson. Literally, if you learn that one mark scheme, any question that you'll be asked, you can answer it. So co-transport. What we're dealing with is we have this normally in our body. Okay, so if we think about it, if we think about our intestines, co-transport occurs. You have your ileum lumen. Okay, this is basically the inside of your intestines. Okay, the inside space. Outside of your intestines, on the outer surface of your intestines, you have epithelial cells. Okay, so if we think about the small intestine as a long tube like this, okay, obviously it's very squashed up in reality, but let's say that's your small intestine. Okay, and this is the inside. The shaded part is the inside of your small intestine, like that. This is your ileum. The inside part is your ileum. Okay, and then the outer layer of the small intestine is your epithelial cells. So on the outer layer over here, I've drawn them bigger, but basically the outer layer is made up of epithelial cells. Okay, it's made up of epithelial cells. The outer layer of the small intestine is made up of epithelial cells. So these, I know if I've drawn them as lines, but basically you need to know that the outer layer is made up of epithelial cells. And basically outside of the small intestine, you have your normal blood supply. Okay, this is your normal blood supply. So this example that co-transport, it normally happens in your small intestines. So what happens is the purpose of this is we need to absorb the glucose from our small intestines into our capillaries, into our blood supply. Okay, so we have our glucose, which is in our small intestine. So think about it, if we eat something, that's going to pass down into our stomach and then it's going to pass into our small intestines. Okay, when it passes into our small intestine, we need to absorb some of the food that we eat. Okay, we can't just excrete it all, we need to absorb some of the food. And glucose definitely needs to be absorbed because glucose is your energy source. If you don't have glucose, then you're not going to be able to respire, you're not going to be able to release energy for active transport. There's loads of things which you can't do if you don't have specific vital supplies in your body. So once it's gone into the small intestine, nutrients are absorbed. And normally what you will do is you will absorb your glucose and you will absorb your amino acids. In this example, we're looking at glucose, but you could also be asked about amino acids. So at the moment, we have our ileum lumen, which is our small intestine in our small intestine. And we need to absorb the glucose all the way from our small intestine through the epithelial cell and into our capillary. How's that going to happen? That's why we have co-transport. So first of all, we have sodium ions and we have potassium ions. So sodium ions are shown over here in dark blue. And what's going to happen is that these sodium ions over here, they're going to move into the capillary from the epithelial cell by active transport using ATP. That's the first step. So sodium ions are actively transported out of the epithelial cell into the blood capillary. At the same time, potassium ions are transported from the capillary into the epithelial cell. You come across this more later on, but this is basically your sodium potassium pump. What's going to happen is that your sodium is going to move from your epithelial cell into the capillary by using active transport. Okay, and that's going to decrease the sodium ion concentration over here because we're actively transporting the sodium ions from the epithelial cell into the capillary. That's going to cause a decrease in sodium ion concentration in your epithelial cell. Okay, so your sodium ion concentration in the epithelial cell is going to be reduced. So what's going to happen is that the sodium ions are going to diffuse from the lumen down the concentration gradient into the epithelial cell by facilitated diffusion. If we have a look at that over here, so we've decreased the sodium ion concentration in the epithelial cell. Because we've decreased that sodium ion concentration, sodium ions over here, which are present in the lumen, a concentration gradient has been established because we've lowered the concentration gradient in the epithelial cell. So these sodium ions can move into the epithelial cell down the concentration gradient because there's a high concentration of sodium ions in the lumen. So sodium ions can now move by facilitated diffusion from the lumen into the epithelial cell. Okay, and they do that by facilitated diffusion. Okay, now when they move by facilitated diffusion into the epithelial cell, what's also going to happen is that they can take a molecule with them. They can take glucose with them, they can take amino acids with them, they can take another molecule with them, attached to them, and they can take it through into the epithelial cell with them. So what they're going to do over here, so what have we done so far? We've got rid of some of the sodium ions in the epithelial cell by active transport, and we've moved them into the capillary. Okay, and that's lowered the concentration of sodium ions in the epithelial cell. What's going to happen now is that the sodium ions from the lumen are going to move by facilitated diffusion into the epithelial cell because there's a high concentration over here and there's a low concentration over here. So it's going to move down the concentration by your facilitated diffusion, and they're not going to move by themselves. They're going to take a molecule of glucose with them. Okay, so we had our sodium ions. They're going to move into the epithelial cell by facilitated diffusion, but they're not going to move alone. They're going to move with a molecule of glucose or with a molecule of amino acids. So we've moved, this glucose is going to be able to move into the epithelial cell as well. Okay, so we've done step one. We've moved the glucose from the small intestine into the epithelial cell. So we're close to moving it into the blood. Okay, so we get glucose over here. Glucose has moved into the epithelial cell. And now what happens is that glucose then moves by facilitated diffusion from the epithelial cell into the blood. So over here, we've brought the glucose into the epithelial cell. Now the glucose can move from a high concentration in the epithelial cell to a low concentration in the capillary by facilitated diffusion. Okay, and then it can be carried away by the capillary to be stored in certain places for energy. That's why it's really important to have this process of co-transport, because if we don't have this process of co-transport, then we're not going to be able to absorb the glucose from our small intestines. So just going over it, in our epithelial cell, we have sodium ions. Okay, we have sodium ions. Those sodium ions are going to be actively transported into the capillary to reduce the sodium ion concentration in the epithelial cell. When that sodium ion concentration has been decreased, that's going to establish a concentration gradient. Okay, because you normally have a high concentration of sodium ions in your lumen. So that concentration of sodium ions, there's a high concentration in the lumen, and now there's a low concentration in the epithelial cell. So the sodium ions are going to move by facilitated diffusion from the lumen into the epithelial cell. And they're not going to go by themselves. They're going to go through a transporter protein, a protein transporter, and they're going to take a molecule of glucose with them. Okay, or they're going to take a molecule of amino acid with them. Okay, so they're going to transport glucose from the lumen into the epithelial cell this way. Once the glucose has reached the epithelial cell, there's a high concentration of glucose in the epithelial cell. Now it needs to be absorbed into our bloodstream so it can be stored in certain places. And because there's a high concentration of glucose over here, that can move by facilitated diffusion into the blood, and the blood will carry the glucose away to wherever it needs to go. Okay, and that's the point of your capillary. So that's your process of co-transport. Okay, just some quick points I need to add on to that. The reason why over here, if we look at the image, glucose is able to move by facilitated diffusion is basically because the blood is constantly moving the glucose. Okay, the blood capillary is constantly moving the glucose to where it needs to be. There's a fast blood supply. It's constantly moving as soon as it gets glucose. So the glucose concentration is always going to be low over here because the glucose will always be transported away. The glucose will always be transported away. It will constantly move and it will go to wherever it needs to go. In that initial point in time, it's going to have a low concentration of glucose. That's why glucose is able to move by facilitated diffusion. And another point, final point to mention, is that on the epithelial cell, you also have microvilli. Okay, and the reason you've looked at this at GCSE, the microvilli, they increase the surface area for co-transport to take place. Okay, because remember, we need as much surface area as we can in the small intestine. Okay, the small intestine is adapted to have a high surface area, because if it doesn't have a high surface area, then absorption is not going to take place at its maximum rate. We want it to take place at its maximum. Okay, that's why you have these villi and you have microvilli. They are there to increase the surface area. So this process of co-transport can occur at a much greater rate. That's the purpose of these microvilli and villi. It's to increase the surface area for co-transport to take place. Okay, and again, they are present on epithelial cells. They are small structures which massively increase your surface area. Okay. So that's all of co-transport. That's basically the end of this unit, and that's everything that you need to know for this unit. Okay, so you've got the method of co-transport there. Sodium ions actively transported into your capillary. That decreases the concentration of sodium ions in your epithelial cell. Sodium ions move by facilitated diffusion from the lumen into the epithelial cell. They take a molecule of glucose or amino acid with them. Okay, glucose concentration in epithelial cell increases, and then glucose moves by facilitated diffusion into blood capillary, where it is transported into certain places to act as a store of energy. Okay, so that is your co-transport process. Okay, and that is everything for cell membranes. It's a short topic, it's not a big topic at all. We covered all the first parts in our last lesson, and today we've looked at active transport and co-transport. It says cells may be adapted for rapid transport across the internal external membranes by an increase in surface area, or by an increase in number of protein channels or carrier molecules in the membrane. So increase in surface area, if they have villi, microvilli, that will increase their surface area for rapid absorption of these molecules across the plasma membrane. And they can also have, in certain places, you can have more protein channels and more carrier molecules. And because you have more protein channels, more carrier molecules, none of the sites will be occupied. Okay? None of the sites will be occupied. And if none of them are occupied, then absorption can keep taking place across the membrane. So if you have a large number of protein channels or carrier molecules, that will always increase the absorption that is taking place. Okay, explain the adaptations of specialized cells in relation to the rate of transport across the internal and external membranes. So this is mostly talking about your villi and your microvilli, which we have looked at. Don't forget that you looked at that GCSE and you also look at it in A level. It's not explicitly covered. However, you will be able to link them to exam questions. Explain how surface area, number of channel or carrier proteins, and difference in gradients of concentration of water potential affect the rate of movement across cell membrane. Again, if you have a higher surface area, a greater number of channel carrier proteins, a greater concentration gradient, a greater water potential difference, that will always increase the rate of movement across your cell membrane.